Hello everyone. In this video series, we are talking about different type of antigen-antibody interactions, and well-known example is precipitation and agglutination. Okay. In case of precipitation, a soluble non-particulate antigen combine with its antibody to produce visible precipitin complex or visible lattice, and this precipitation reaction only occur in presence of electrolyte like NaCl at optimum temperature and pH. Okay, so the antigen antibody reaction of precipitation type can occur in liquid as well as in gel medium. Okay, suppose first we take liquid medium precipitation, then the example is precipitin ring test. Okay, so precipitin ring test is the type of a precipitation reaction which occur in liquid medium. Antigen and antibody combine or get precipitate in the liquid medium. So this is the first example. Now the next is if the same reaction occur in the gel medium, then it is called as diffusion. And the examples of gel medium or the diffusion is radial immunodiffusion. It is also called as single radial immunodiffusion, then double immunodiffusion, third immunoelectrophoresis. So we have already talked about precipitation, ring test, radial immunodiffusion and double immunodiffusion in last few videos. Now in this lecture, I'm going to focus on immunoelectrophoresis. Okay, name itself suggests it is the type of immune reaction in which we are going to use electricity, okay, or electrophoresis procedure. Now remember that in case of radial immunodiffusion or single radial immunodiffusion, only antigen move towards antibody while antibody remains stationary in the gel. So we call it as a single radial immunodiffusion. Now in case of double immunodiffusion or Auchterlonie test, both antigen and antibody are free in the gel, they can move towards one another. So we call it as a double immunodiffusion. Now in case of immunoelectrophoresis, first we are going to do the electrophoresis for separation of the component of the antigen mixture, and after electrophoresis we shift towards double immunodiffusion. That means in case of immunoelectrophoresis, two-stage procedure is followed. Number one, or first step of the immunoelectrophoresis is separation of antigen, separation of antigen mixture by electrophoresis or by using electricity. And the second stage is after separation of the antigen, we move towards double immunodiffusion. Then we do double immunodiffusion. So the immunoelectrophoresis technique is the mixture of, or it is the combination of electrophoresis as well as double immunodiffusion technique. Okay, the immunoelectrophoresis was first time reported in 1953 by Graber and Williams. Okay, so these two scientists first time report immunoelectrophoresis technique. It is very sensitive technique because we are combining two different procedure together. First is electrophoresis and second is double immunodiffusion. Okay, so when we compare immunoelectrophoresis with all other different type of precipitation reaction, the sensitivity will going to increase, or sensitivity always remain high of immunoelectrophoresis. Okay, the technique have ability to separate individual antigen or individual protein from the mixture of protein which generally present in the serum. The technique have very high value in the diagnostic. Okay, you can easily find out different type of proteins or different type of antigen present in the given sample. Okay, so First, we will going to discuss about principle of this technique. Okay, so first, the technique use electric current or electricity to resolve antigen. Okay, so we know that antigens are protein, and protein may carry charge, either positive or negative, and in this technique we are going to use agarose or agar gel, and in the gel we are going to make a well, and inside that well we are going to add mixture of the antigen, and each antigen which is present in the mixture carry different charges, and when we apply electric current onto the gel, this antigen will going to migrate towards different pole, either towards anode or towards cathode, depending upon their charge and depending upon their size. Okay, so the separation of the antigen occur in the electrophoresis or in the first step by size and charge. Okay, there are various other factors which also play important role in the electrophoresis separation of the antigen, but the two most important are size of the antigen and charge. Okay, if the antigen is larger, then the movement of the antigen through the gel will be very slow. Okay, and if the size of the antigen is small, it can migrate fast through the pores of the gel. Similarly, charge also going to affect the final mobility of the antigen through the gel. So these are the two important factors which affect electrophoretic mobility of the antigen or protein through the gel. And once we apply the electric current, we will going to separate the antigen in the reaction mixture in the gel. And then next step is we are going to add addition of specific antibodies. Okay, and now this antigen and antibody migrate towards one another in the gel, and this is called as double immunodiffusion, and due to this migration a zone of equivalence reach in the gel which will going to produce lattice. Okay, zone of equivalence lead to lattice formation. Okay, antigen antibody molecule combine with one another, a network is produced which get precipitate out in the gel, and this type of antigen antibody network is called as lattice. Okay, and once lattice is produced, the white precipitation like line become visible in the gel. So this is the basic principle of the test, that first we are going to separate the different components or different antigen from the sample, and then we are going to do double immunodiffusion. Okay, so for this test, various things one should need in the lab. Okay, so the requirements are: number one, agarose powder. Okay, agarose or agar, or you can use polyacrylamide powder. Then the second is assay buffer, because it is an immunological reaction. It contains antigen and antibody, in both are proteins. They require optimum pH for their activity. So we have to use assay buffer. We cannot use normal distilled water for preparation of different gel and antigen antibody mixtures. Okay, because buffer will going to maintain the pH constant, which will going to keep the antigen and antibody intact. Okay, the next thing that you need is mixture of antigen, then antibody specific for that particular antigen, well puncture, okay, then gel cutter and other type of requirements like glassware, conical flask, beaker, okay, glass slide, moist chamber. So all these are the requirements which one should need to perform immunoelectrophoresis. Along with this, you also need electrophoresis unit. Okay, so now I'm going to discuss the procedure Procedure of this test, that is immunoelectrophoresis. First step is preparation of 1% agarose gel. Okay, and in my last video I have discussed in detail, to prepare 1% agarose gel you have to add 1 gram agarose in 100 ml as a buffer. Okay, so you have to weigh 1 gram agarose and add in the 100 ml as a buffer, and to dissolve it, the next step is boiling of gel to dissolve. Okay, and at the time of boiling, the agarose get dissolved in the buffer to produce clear solution. And after this, the next step is allow gel to cool up to 55 to 60 degrees Celsius. Okay, and after that you can pour this gel on glass slide. Okay, suppose this is your glass slide, clean, grease-free glass slide, and now you have make 1% agarose gel, you have boiled it, completely dissolved it, and then you are going to just pour that agarose gel onto the glass slide. Okay, it will going to make a layer, thick layer of the agarose onto this glass slide. Okay, next step is allowed it to solidify for 30 minutes, and after solidification of this agarose gel onto the glass slide, next step is making the well. For example, I have made the well at this point, okay, with the help of gel puncture. Okay, so the next step is making well for antigen mixture. Okay, so after preparation of the well or hole in the gel, next step is you have to add your sample or mixture of the antigen. Okay, you have to add this antigen mixture into the well with the help of micropipette, and after addition of this antigen into the well, the next step is electrophoresis. Okay, then we are going to use electrophoresis unit. We have to place this glass slide onto the electrophoresis chamber, and then we have to just apply the electric current approximately 100 volt for 30 minutes. Okay, now we know that in electrophoresis tank we are going to have one positive and another negative electrode. Okay, that is cathode and anode. Okay, now if the antigen mixture contain, for example, 4 antigen A, B, C and D, okay, out of which this A and B have positive charge and C and D have negative charge, and we know that if the electricity is applied, now this negatively charged C and D antigen will going to move towards positively charged electrode. Okay, that is anode, and positively charged antigen like A and B will going to move towards negatively charged electrode, that is cathode. Okay, so these are the electrodes, negative electrode and positive electrode. So in this 30 minutes of electrophoresis, the movement of the different antigen from the mixture will going to start towards both the electrodes depending upon their charge. Another factor come into the picture here, which is size. Now suppose this C is large than D, and A is large than B in size. So in this case, this A and B will going to migrate towards right side, towards negative electrode, and A is larger, so A will going to move slower than B. So here you can see B move fast, okay, from well towards electrode, so it cover maximum distance followed by A. Similarly, towards positive electrode, this C and D antigen will going to migrate, out of which C antigen have large size, so C antigen face more resistance from the gel for migration, so it migrates slowly. So D antigen will going to reach maximum distance from the well in this direction. Okay, so you will going to get 4 different type of positions where antigens are present now, like A and B towards negative electrode and C and D towards positive electrode. Now the next step, this is the first 7 steps which end the electrophoresis process. Now the next stage is double immunodiffusion. In double immunodiffusion, we are going to cut the strip or cut the trot in the gel. Okay, so parallel to this migration line, we are going to cut the gel with the help of gel cutter like this. We are going to take thin slice of the gel like this, and this will always be parallel towards the antigen migration line. And then we're going to add, next step is addition of antibodies. Now we're going to add antibody specific for this antigen into this plate. Okay, and once antibody is added, then we're going to keep this glass plate for incubation at 37 degrees Celsius for 24 hours, 24 to 48 hours in moist chamber. Because it is an antigen-antibody reaction which require moisture, the gel may get dry during the incubation, so we have to maintain the moisture, and for that we have to keep this glass plate in a chamber for maintaining moisture, and for that we can use cotton balls soaked in the glycerol. Okay, now during the incubation, what will going to happen? The antibody migrate towards antigen, and antigen also start migrating towards antibody. So in this case, both antigen and antibody are free. They are migrating towards one another. So we call it as a double immunodiffusion. Okay, and once they reach or once they interact with one another, zone of equivalence produce that ultimately form lattice. Okay, and the lattice will going to give line of precipitation like this. So depending upon size or intensity, position and shape, okay, so these 3 factors come into the picture now: size of the line, then intensity of the line, and the position of the line on the gel. You can easily make conclusion about different type of antigen present in the mixture. Initially I was unaware about the different type of antigen present in the mixture, but now I can say that in this antigen mixture there are 4 different antigens are present, like 1, 2, 3 and 4, because I have got 4 line of precipitation. And depending upon the shape of the precipitation line or its intensity, we can finally make conclusion about the concentration of each type of antigen present in the mixture, as well as we can conclude what type of charges the antigen having in the mixture. Okay, so this is the technique which is frequently used for separation of different antigen from the reaction mixture. Okay, in this way we have finished with the precipitation reaction Okay, and it's different type like precipitation ring test, radial immunodiffusion, double immunodiffusion, and immunoelectrophoresis. Another variation of the immunoelectrophoresis is rocket immunoelectrophoresis. In my next video, I'm going to discuss about it. Thank you.